In this script, we will explore the intricate details surrounding the regulation of fatty acid synthesis. Fatty acids play a fundamental role in various physiological processes, including energy storage, membrane structure, and cellular signaling. The tight regulation of fatty acid synthesis is essential for maintaining cellular homeostasis and preventing metabolic disorders such as obesity and diabetes. Fatty acid synthesis is regulated by various transcription factors, and PERs are a key group of regulators. PERs are nuclear receptors that bind to specific DNA sequences, known as PER response elements, on target genes. Upon activation by fatty acids or synthetic agonists, PERs form heterodimers with retinoid X receptors and bind to these response elements, initiating the transcription of genes involved in fatty acid synthesis. PER alpha, PER beta, delta, and PER gamma are the three isoforms of PERs, each with distinct tissue distribution and functional roles. PER alpha promotes fatty acid oxidation, whereas PER gamma is mainly involved in adipogenesis and lipid storage. PER beta, delta is less understood but has been implicated in regulating fatty acid metabolism in various tissues. Acetyl-CoA carboxylase, ACC, is a key enzyme in fatty acid synthesis, catalyzing the conversion of acetyl-CoA to malonyl-CoA, the building block of fatty acids. ACC exists as two isoforms, ACC1 and ACC2, with distinct subcellular localizations and functions. ACC1 is primarily responsible for cytosolic malonyl-CoA synthesis, while ACC2 is located in the mitochondria and regulates fatty acid oxidation by controlling malonyl-CoA levels. The activity of ACC is tightly regulated by reversible phosphorylation events. Phosphorylation of ACC by AMP-activated protein kinase, AMPK, inhibits its activity, leading to decreased malonyl-CoA production and increased fatty acid oxidation. Conversely, dephosphorylation of ACC by protein phosphatases, such as protein phosphatase 2A, PP2A, promotes its activity, resulting in elevated malonyl-CoA levels and enhanced fatty acid synthesis. This dynamic phosphorylation-dephosphorylation cycle serves as a crucial regulatory mechanism for balancing fatty acid synthesis and oxidation. During fatty acid synthesis, acyl carrier protein, ACP, plays a crucial role in shuttling intermediates between the different enzymatic activities. ACP is a small protein that interacts with several enzymes involved in fatty acid synthesis, including ketoacyl synthase, ketoacyl reductase, and enol reductase. ACP achieves this by covalently binding the growing fatty acid chain during synthesis, forming a thioester linkage with phosphopontate HYNE, a cofactor derived from coenzyme A, CoA. This allows the transfer of the fatty acid intermediates between the various enzymatic sites, facilitating the elongation process. The activity of ACP is regulated by several factors, including phosphorylation and protein-protein interactions. Phosphorylation of ACP by protein kinases, such as pKa, modulates its binding affinity to the fatty acid synthesis enzymes, thereby influencing the overall rate of fatty acid synthesis. Fatty acid synthase, FAS, is a key enzyme in the de novo synthesis of fatty acids. The expression of FAS is tightly regulated at the transcriptional level by various transcription factors, including sterol regulatory element binding protein 1C, SREP1C, and carbohydrate responsive element binding protein, CHREP. SREP1C is primarily regulated by insulin signaling and is activated in response to high carbohydrate or insulin levels. Upon activation, SREP1C translocates to the nucleus, where it binds to sterol regulatory element, SRE, sequences in the promoters of target genes, including FAS. This binding activates the transcription of FAS and other lipogenic genes, promoting fatty acid synthesis. CHREP, on the other hand, is activated by glucose and acts as a glucose sensor in the liver. It induces the transcription of FAS and other lipogenic genes by binding to carbohydrate responsive element, CHO-RE, sequences in their promoters. This ensures that fatty acid synthesis is coordinated with the availability of glucose as a substrate. The malonyl-CoA/carnitine shuttle plays a vital role in coordinating fatty acid synthesis and beta oxidation, two opposing metabolic pathways that occur in different cellular compartments. Malonyl-CoA, the product of ACC-mediated fatty acid synthesis, acts as an allosteric inhibitor of carnitine palmitoyltransferase 1, CPT1, a key enzyme in the transport of fatty acids into the mitochondria for beta oxidation. By inhibiting CPT1, malonyl-CoA prevents the simultaneous occurrence of fatty acid synthesis and oxidation in the same cellular compartment. This spatial segregation achieved by malonyl-CoA ensures the efficient production of fatty acids without compromising beta oxidation. As cellular energy demands change, the levels of melanyl-CoA dynamically fluctuate, allowing for the appropriate coordination between fatty acid synthesis and beta oxidation. Fatty acid synthesis is particularly important in lipogenic tissues, such as the liver and adipose tissue, where excess nutrients are stored as triglycerides for energy reserve. In the liver, insulin plays a significant role in promoting both glucose uptake and fatty acid synthesis. Insulin activates key lipogenic enzymes, including ACC, Phos, and glycerol 3-phosphate acetyltransferase, GPAT, through various mechanisms involving protein phosphorylation and dephosphorylation events. In adipose tissue, fatty acid synthesis is primarily regulated by the availability of glucose and adipogenic transcription factors, such as pergamma. Adipocytes convert excess glucose into fatty acids through de novo lipogenesis, which supports triglyceride storage within adipose tissue. Sterol regulatory element binding proteins, SREBs, play a crucial role in controlling fatty acid synthesis by regulating the expression of key lipogenic genes. SREBs are transcription factors that are synthesized as inactive precursors in the endoplasmic reticulum, membrane. Upon nutritional and hormonal cues, including insulin and low cellular sterol levels, SREBs are proteolytically cleaved, releasing the active transcription factor fragment. These active SREBs translocate to the nucleus where they bind to SRE sequences in the promoters of lipogenic genes, such as Phos and ACC. This binding promotes the transcription of these genes and facilitates the synthesis of fatty acids, ensuring an adequate supply of lipids for cellular functions. The mechanistic target of rapamycin, mTOR, signaling pathway plays a crucial role in regulating lipogenesis and fatty acid synthesis in response to various stimuli, including growth factors, nutrients, and energy status. mTOR exists in two complexes, mTOR complex 1, mTORC1, and mTOR complex 2, mTORC2. mTORC1 integrates signals from growth factors and nutrients to promote lipogenesis and fatty acid synthesis by phosphorylating and inhibiting the activity of inhibitory proteins, such as tuberous sclerosis complex 2, TSC2, and eukaryotic translation initiation factor 4E binding protein 1, 4EBP1. mTORC1 activation enhances the expression of lipogenic enzymes, including ACC and Phos, promoting de novo fatty acid synthesis. Furthermore, mTORC1 regulates the expression and activity of SREBs, further contributing to lipogenesis. Dietary factors and the nutritional status of an organism play a significant role in modulating fatty acid synthesis. High-carbohydrate diets stimulate insulin secretion, which, in turn, activates lipogenic transcription factors, such as SREB1C and CHREAP. These transcription factors, along with pergamma, promote the expression of genes involved in fatty acid synthesis, leading to increased production of fatty acids. Conversely, a low-carbohydrate, high-fat diet, such as a ketogenic diet, suppresses insulin signaling and promotes fatty acid oxidation. This shift in substrate availability and hormonal signaling enhances fatty acid oxidation while suppressing de novo lipogenesis. Furthermore, the balance between macronutrients, such as the ratio of carbohydrates to fats, also influences the rate of fatty acid synthesis. Excessive calorie intake, particularly from carbohydrates and saturated fats, can lead to dysregulated fatty acid synthesis and metabolic diseases. Cancer cells have unique metabolic pathways that support their rapid growth. One such pathway is fatty acid synthesis, which is upregulated in many types of cancer. This excessive lipid production provides the energy and building blocks needed for tumor progression. In addition, fatty acid synthesis generates lipids for membrane formation and signaling molecules. By targeting this pathway, specifically by inhibiting key enzymes involved in lipogenesis, we may be able to develop new therapies to slow down tumor growth. Preclinical and early clinical studies have shown promising results in this area. Fatty acid synthesis does not occur in isolation but rather interacts and crosstalks with other metabolic pathways, including glycolysis and the tricarboxylic acid, TCA, cycle. Glycolysis provides the precursor molecule, acetyl-CoA, for fatty acid synthesis. Glucose is converted into pyruvate through glycolysis, and pyruvate is subsequently converted into acetyl-CoA, which becomes the building block for fatty acid synthesis. Conversely, fatty acid oxidation generates acetyl-CoA, which feeds into the TCA cycle. This acetyl-CoA can be further utilized for the production of energy or diverted towards fatty acid synthesis, depending on the cellular energy demands and metabolic state. The intricate coordination and regulation of these metabolic pathways ensure the appropriate balance between energy production and storage, allowing cells to adapt to fluctuating nutrient availability and metabolic demands. Emerging evidence suggests that microRNAs, miRNAs, and epigenetic factors play critical roles in the regulation of fatty acid synthesis. miRNAs are small non-coding RNAs that post-transcriptionally regulate gene expression by binding to the target mRNA and inhibiting its translation or promoting its degradation. Several miRNAs have been identified as regulators of lipogenic genes, such as FOS and ACC, thereby modulating fatty acid synthesis. Furthermore, epigenetic modifications, including DNA methylation and histone modifications, can govern the expression of lipogenic genes. Aberrant DNA methylation patterns or altered histone acetylation and methylation states can dysregulate fatty acid synthesis and contribute to the development of metabolic disorders. Understanding the role of miRNAs and epigenetic factors in fatty acid synthesis regulation provides novel insights into the intricate mechanisms that govern this metabolic pathway. In conclusion, the process of regulating fatty acid synthesis is complex and controlled by various factors and pathways. Transcription factors like PERS, SREBS, and CH-REAP initiate gene expression for lipid production. Enzymes such as ACC, FOS, and ACP govern the biochemical reactions needed for fatty acid synthesis. Signaling pathways like mTOR and dietary factors influence the overall synthesis rate. Dysregulation can cause metabolic disorders like obesity and cancer. Understanding this regulation offers insights for potential therapies in metabolic diseases.